Hello everybody, I am Amel Necibi, a PhD student. I'm going to present my article titled Assessment of Epileptic Gamma Oscillations Networks Connectivity. I have worked with Mrs. Abir Hadriche and Mrs. Nawel Jmail. First of all, I will start my work by the plan, which contain introduction, methods, metrics of network connectivities, results, and I achieved with a conclusion. Although the technology has shown significant development until today, studying the brain is still quite a complex task. The brain designates the most important part of the central nervous system. It's the first responsible for intellectual and cognitive functions. Thus, the regulation of vital functions is formed by two hemispheres, one right and one left. Each of them is responsible of the reverse region, right hemispheres for the side and left hemispheres for the right side. There are several neurological diseases that overwhelm the human brain. Among these pathologies, we find Alzheimer, brain tumor, Parkinson, and epilepsy. In our work, we will be interested in epilepsy. Epilepsy Epilepsy is a simultaneous electrical discharge for a large population of neurons. Epilepsy or an epileptic seizure is an abnormal electrical discharge from neurons in the cerebral cortex. These symptoms are repetitive seizure. The seizure is started by a partial crisis and spread of the crisis, final generalized of the crisis, and we have here a crisis of epilepsy. The main techniques of acquiring brain activity, either non-invasive techniques such as EEG and MEG, or invasive techniques such as intracerebral EEG, IEEG. In my work, I use the non-invasive technique magnetoencephalography MEG. MEG is used to determine cortical regions of the brain and to look up epileptogenic regions. The advantage of MEG over surface EEG is that the magnetic field is not influenced by the successive layers in pass through, unlike EEG. We applied a pre-processing chain to assess the rate of epileptic gamma oscillation connectivity among meg of each technique. We proposed here to rate the performance of issued inverse problem in identifying epileptic zone. We pass now to the methods. Then we used four methods for inverse problem to explain cortical areas and neuron generators of excessive discharges. MNE, based on the regularization of means which can have an effect on the estimation of time series in the form of crosstalk between the sources. DSPM offers more interest to deeper areas. Accelerator allows to compensate for the depth base by smooth maps whose regions are all connected regardless of the depth of the source. Finally, WMEM, based on wavelet, can also be applied in wavelet representation. We pass now to the metrics of network connectivity that I used to assess the connectivity between neurons. We have the first one is correlation, is a relatively simple non-directed connectivity metric of association between time series. Then covariance is a complex valued metric that measures linear relationship of two signals in the frequency domain. Then gradual causality is a measure of directed functional connectivity based on the Wiener gradual causality framework. Gradual causality measure linear dependencies between time series and prediction tests of the signal future. Finally, we have PLV, phase looking value, is a popular metric defined as the average vector of many unit vectors length whose phase angle corresponds to the phase difference between two time series. We pass now to the materials that are used for the process. The pre-processing steps revealed in this project are carried out by the MATLAB software and Brainstorm. We pass now to the results of assessment of epileptic gamma oscillations network connectivity. Is shown in figure 1 that we describe active region, local peaks of source activation film of pure oscillationist activities using MNE, DSPM, SLURITA, and WMEM inverse problem methods. SLURITA shows the highest number of active regions ROI, region of interest. The lowest active regions are obtained by WMEM. We pass now to the the connectivity between neurons, the network connectivity. We have first metric is correlation. In figure 2, the SPM indicate a strong correlation between our selected regions. Meantime, Esloretta and WMM show an average correlation. For MNE method, all selected active regions exhibit weak correlation. Pass now to covariance. In covariance, that figure 3, we note that for DSPM, SLORITA, and WMEM methods have almost an equivalent covariance degree. Moreover, MNE show a stronger connectivity. Granger causality in figure 4, connectivity is depicted by a link with direction in or out. Thanks to this metric specification is shown in the figure. We obtained a measure of directed functional connectivity. WMEM shows strong connectivity between region of interest, and DSPM represent weak connection. Finally, we have PLV fast locking value in figure 5. MNE and Sloretta represent few links but with a high connection. For DSPM and WMEM, they have more links but weak connection. We shift by the conclusion. WMEM, wavelet maximum entropy on the mean, had the highest number of links that are matching between MEG network connectivity based on correlation, covariance, gradual causality, and fast looking value between active sources, followed by the SPM, Astrolita, and MNE. The acquired results of these methods are generally consistent with the neurologist's diagnosis. In fact, all of the techniques used were able to locate with good precision the active sources of the gamma oscillation MEG epileptic events. Thank you for your attention.